So I would like to welcome everybody. We're pleased that our general clubs are back and even more excited about the TFP that is around the corner. We're all meeting up next week. But before we start with our speakers, I would briefly go through the usual ground rules, as we do every time. So I would like to ask you to turn your microphones off and use the chat button or raise your virtual hand to address your questions to the speakers, preferably at the end of the talk or in between the talk if it's necessary for your understanding. Finally, we would like to thank our speakers for being here today and accepting the invitation. Also, Dr. Vatakis, Dr. Weiner, Dr. Teki, and the ECR committee for putting these meetings in place. So we will start today with Dr. Ashley Schormans, who is a postdoctoral associate and BrainsCAN postdoctoral fellow at Western University. She obtained her PhD in anatomy and cell biology at Western University in 2019, where her research focused on investigating cortical cross-modal plasticity following adult onset noise-induced hearing loss, as well as developing novel rodent models of audiovisual temporal perception. As a postdoc, she has focused on uncovering the mechanisms by which inhibitory neurotransmission and top-down circuits finally control audiovisual perception using a combination of translational rodent behavioural models, optogenetics, and in vivo electrophysiology. So today, Ashley will talk about the investigation of the neural basis of audiovisual temporal processing and perception in rodents. So, Ashley, the floor is yours, and we're all ears.
Right, great. So thank you very much for the invitation and for inviting me to talk. So today I'm going to be telling you some of the work that I've been doing with my lab over the last few years towards the goal of investigating the neural basis of audiovisual temporal processing and perception, specifically using rodents as our model. So on a daily basis, our brains naturally merge information from multiple sensory modalities, and stimuli that are spatially and temporally coordinate. Stimuli that are spatially and temporally coordinate tend to influence one another's processing, where stimuli that are discoordinate in space and time tend not to influence one another. Experimentally, this has been studied extensively using multisensory illusions, such as the sound-induced flash illusion, as well as perceptual judgment tasks, such as the temporal order judgment task and the synchrony judgment task. However, until recently, no rodent models of either behavioral paradigm existed. To that end, our lab and others have begun to develop rodent models of audiovisual temporal perception in order to investigate whether or not rats show the ability to perceive the relative timing of audiovisual stimuli. Our lab has decided to focus on the temporal order judgment task, where we train rats to determine whether or not an auditory or a visual stimulus was presented first. To do this, we use two alternative forced choice tasks and appetitive conditioning in order to train the rats to associate a given feed or trough with a given stimulus. This is just a short video showing how we're able to train the rats to do this. So our operant chambers consist of a left feeder trough, a right feeder trough, and a center nose poke. Once rats are placed in the behavioral chamber, they can then initiate a trial by holding their nose in the center nose poke, after which the stimulus will be played. If it's a visual first stimulus, they go to the right feeder trough. And if it's an auditory first stimulus, they go to the left. Upon doing a correct trial, they will receive a food pellet as a reward. And if they get it incorrect, they get a punishment, which consists of the house lights turning off and an inability to complete a new trial for about 15 seconds. So once rats are fully trained on this task and they're able to distinguish stimuli, we can then introduce experimental test sessions where rats are introduced with novel SOAs, and SOAs is stimulus onset asynchrony, so the timing between the stimuli. So typically rats in the presentation I'll be showing you today are trained down to an SOA of about 200 milliseconds. So visual first 200 or auditory first 200, and then we introduce SOAs between those stimuli. So in this case, 0, plus or minus 40, and plus or minus 100. It's important to note that when these novel stimuli are presented, the rats are neither punished nor rewarded. So we're truly getting an indication of what they perceive. So if we plot a curve of what it looks like when rats perform these tasks, along the x-axis here, laser pointer. Along the x-axis here, we have our point of subjective simultaneity. So the negative numbers represent auditory first stimuli, and positive numbers represent visual first stimuli. And then we have the proportion of responses perceived as visuals first. So if it's an auditory first trial at 200 milliseconds, so minus 200, they should not be going to the visual first side. So this number should be low. And then the opposite occurs when we have a visual leading stimulus that they are well trained at. So this should be high. And then our novel SOAs are in between, and you can see we get this nice sigmoidal curve. We can extract a few metrics from this curve, such as the point of subjective simultaneity, which describes the actual timing of the audiovisual stimuli when the observer is most unsure of the temporal order. We can also extract the just noticeable difference, which represents the smallest interval between the separately presented auditory and visual stimuli that can be reliably detected. So while we know that rats can give metrics that are fairly consistent with humans, we sought to further confirm whether rodents actually demonstrate some of the hallmarks of audiovisual temporal perception, specifically using the TOJ task. So some of the hallmarks that we're interested in investigating are whether or not they show sensitivity to experimental conditions, such as stimulus duration and stimulus intensity. Do they show malleability to previous sensory experience? So do they show effects of previous sensory exposure? We also want to determine whether or not there were shifts in perception based off the previous trial type. So does rapid recalibration or the previous trial influence rats' perception? So we're first going to start with looking at whether or not rats are sensitive to experimental conditions. So the first condition we looked at was sound intensity. So if we start with our normal training intensity, so rats are typically trained with an acoustic stimulus at 75 decibels. You can see we get a nice psychophysical curve, quite consistent with the curve you saw on the previous slide. We extract our PSS in J&D. You can see our PSS is right around 0, and our J&D is hovering around 50 milliseconds, which is quite consistent across rats. What happens if we decrease the intensity going down to 60 dB? You can see we're getting a leftward shift in our PSS. So in order for the stimuli to be perceived as synchronous, the auditory stimulus must be presented farther out front. And we're actually getting a slight increase in our J&D. So we have slight impaired temporal sensitivity at 60 dB. And this is because this left side of the curve, or the auditory side of the curve, is slightly impaired. If we increase our stimulus intensity now and go up to 90 decibels, so it's quite a loud sound, you can see we're getting an opposite pattern occurring, whereby our curve is shifting towards the right. So the PSS is becoming more visual first. However, we're seeing no changes in J&D. So ultimately, this shows that rats are consistent with humans, as stimulus intensity did influence their perception. Next, we'll quickly look at stimulus duration. So if we look at the duration at which they're trained at, so we normally use a 50 millisecond acoustic stimulus. You can see we get a decent psychophysical curve. These SOAs are a little bit farther out because these rats were only trained down to 400 milliseconds, which you can see as their J&D is much larger. Their PSS is sitting slightly on the visual first side. However, that is relatively consistent with rats trained out to this delay. So you can see if we were to draw a line out, it would be on the slightly visual first side. So if we increase our stimulus duration, if we double it up to 100 milliseconds, you can actually see that our PSS is staying quite consistent. The middle of this curve is not moving. However, we do have a decrease in our J&D. So we are showing improved temporal sensitivity, and that can be observed through the slope of this line. So it's much steeper. We then decrease our stimulus duration down to 20 milliseconds, sorry. You can see again, we're getting inconsistent PSS. However, we're actually getting an increase in our J&D, which can be observed through this slope of this line flattening out a little bit. So this shows that the duration of both the auditory and the visual stimulus had relatively no net effect on the perception of simultaneity, but it did affect their temporal sensitivity. So if we quickly look at what happens to perceptual malleability based off prior sensory experience, what we did was passively expose animals to auditory and visual stimuli. So in the first condition we did is we exposed them to synchronous auditory and visual stimuli. So we placed them in an operant chamber. They were in the dark. They were presented with 360 trials of synchronously presented auditory and visual stimuli for 6 minutes, immediately prior to testing. And this is the resultant curve. You can see they're actually quite good at each of their offsets, with our PSS shift sitting right around 0 and our J&D around 40 to 50 milliseconds. So if we introduce a temporal lag in our stimuli during our passive exposure prior to testing, if we introduce an asynchronous stimulus with an auditory leading condition, so in this case, the auditory stimulus is going to precede the visual by 160 milliseconds. And again, they're exposed to that for 6 minutes right before testing. You can actually see we're getting a leftward shift in our PSS. So our PSS is becoming more negative. So it's going more in the auditory first direction. And we're getting no change in our J&D. We look at exposure to a visual leading stimulus. So the same case, just in the opposite pattern, a visual stimulus is going to lead the auditory by 160 milliseconds. We're getting the opposite pattern where our PSS is becoming more positive and we're getting no change in J&D. So this is quite consistent with humans, where we're showing that exposure to asynchronous stimuli prior to experimental testing actually does shift the rat's perception of simultaneity in the direction of the leading stimulus. So the last thing we wanted to confirm is whether or not previous trial affected temporal perception. So if we quickly look at all of the combined trials together, not surprisingly, our PSS and J&D look consistent with previous studies. So now we're going to separate these combined trials, or all trials, into different categories. So we're going to split the trials into ones in which the preceding trial was an auditory leading stimulus. So we're going to take our trial, we're going to go T-1, so the trial before it is that trial auditory first. We're going to pull that into auditory leading trials, and then the opposite with visual leading trials. So if we look at our curve in response to auditory leading trials, you can see we're getting quite a dramatic shift in our PSS. It's one of the strongest phenomenons that we've seen in the rats. So our PSS is becoming more negative, and we are seeing no change in our J&D. We look at visual leading trials, we're getting pulled in the opposite direction again. So we're getting pulled towards the visual first side with no change in J&D. And this is a pretty strong, consistent phenomenon that's observed in the rats, which is also observed in the humans. So rapid recalibration or single trial recalibration is in fact observed in rats. We've seen this in multiple cohorts of rats now, as the previous trial does shift rats' perception in the direction of the preceding trial. So to quickly summarize this section, we saw that consistent with humans, rats do show a shift in PSS based off stimulus intensity, and they show no net effect of stimulus duration on PSS. They do show malleability based off prior sensory experience. They do show single trial or rapid recalibration. So ultimately, we believe that this shows that rats represent a viable model of audiovisual temporal perception, as they do demonstrate some of the key features that are observed in humans. So now that we have a model of audiovisual temporal perception, we plan to use this model in order to investigate what the role of the multisensory cortex is in audiovisual perception, and then examine whether or not an imbalance of excitation and inhibition, or EI, has on the multisensory cortex at the level of processing and perception. So using a combination of in vivo electrophysiology, neuropharmacology, and behavioral testing, we aim to uncover the mechanisms by which inhibitory neurotransmission finally control one's perception. But you might first be asking, so where is the multisensory cortex in the rat? So conserved across species, there are functionally specialized regions of the mammalian cortex that contain neurons that are capable of integrating both auditory and visual systems information. These higher order regions occur at the borders of our primary sensory cortex. So if we look at a lateral view of the rat brain, we can overlay the primary auditory cortex, the primary visual cortex. And the area that we are interested in is the lateral extrastriate visual cortex. And this is the area that is sandwiched right between our primary auditory and visual cortex. If we take a lateral coronal section through our rat brain, we can then look to see what this looks like coronally. So we can see we have visual cortex at the top. As we move more ventrally, we're going to find our multisensory cortex sandwiched in between our visual and then our auditory responses. So now that we've highlighted our area of interest, we're interested in the lateral extrastriate visual cortex, or V2L, or the audiovisual cortex, whatever you want to call it. We wanted to examine what the role of this cortical region was in audiovisual perception. So to do this, we silenced the cortex. And we did this in 8 trained TOJ rats that were chronically implanted with drug infusion cannula into the V2L cortex. So this is just a schematic of what the cannula looked like. And then this is an actual rat with the cannulas. We performed experimental test sessions following an infusion of ACSF, which is our vehicle control, and mucimol, which is a GABA-A agonist, which tends to result in an increase in the level of GABA, essentially turning off a cortical region. So we first wanted to confirm that our vehicle control, so ACSF or artificial cerebrospinal fluid, had no effect on temporal perception. And we can see that once we plot the curves. The curve still shows a nice sigmoidal curve fitting function across all SOAs. So what happens when we silence this cortical region? You can actually see that we're getting a shift towards the right. So our PSS is becoming more positive, which means in order to perceive stimuli as synchronous, this visual stimulus must precede the auditory by a greater lag. And we have right now have no statistical effect on J and D, which shows that the V2L cortex, or the multisensory cortex, plays a role in perceiving the relative timing of the audiovisual stimuli, but does not actually influence the ability to detect the subtle timing differences between them. So now that we know that this cortical region plays a role in perception, what if we manipulate this region and pull the level of excitation and inhibition in a different direction? So to do this, electrocellular recordings were performed in anesthetized rats before and after the local microinfusion of ACSF, which is again our control, and or gabazine, which is a GABA-A antagonist, which means that it competes with GABA and thus decreases the level of inhibition within the cortex. So to do this, a 32-channel electrode was inserted perpendicular to the cortical surface, and local field potentials were recorded across all cortical layers. A one-dimensional CSD analysis, or current source density, was then applied to the local field potentials recorded across the entire cortical circuit. The CSD provides a measure of the total current density that enters or leaves the extracellular space through the cell membrane. And current sinks, such as the granular, infragranular, and supragranular sink, represent the flow of positive ions into the neural tissue from the extracellular space, which represent axonal depolarizations, so activity going on in that area. So if you think of this heat map as a topographic map, with the hills being represented in the red and troughs being in the blue, we can then take a cross section through our map and essentially give us a 2D figure of what's going on within each of the granular layers. So for today, we're just going to focus on what's occurring within the granular layer, so the green one here. But what we can do is then use this 2D map in order to analyze the peak amplitude, so how hot or how active is our current sink, and we can also look at the onset of our activity. So what happens when we play audiovisual stimuli? So if we present synchronous audiovisual stimuli, so stimuli that occur at the same moment in time, you can actually see within the multisensory cortex, they don't arrive at this cortical region at the same time. And this is due to the lag of visual stimuli coming off the retina. We again take our slice through there and look at our 2D image. Again, you can see the same phenomenon. We're getting these two double peaks. If we slew the timing between the stimuli, so we move the visual stimulus 40 milliseconds in front of the auditory, you can see we're getting the single peaked response. So a single sync is occurring in response to those two stimuli being played. So we're only getting one peak. And if we look at the opposite way, so auditory leading by 40 milliseconds, so minus 40 SOA, you can see we're getting two very far distinct peaks occurring in response to both the auditory and the visual stimuli. So what we can do is then quantify the probability of getting a single peaked response. So if we look at positive 40 milliseconds, you can see we have a very high probability of getting a single peaked response pretty much every time. You're only going to get one single peak. The opposite occurs when you're at minus 40 milliseconds. You see we're getting two very distinct peaks, with the trough actually dropping below baseline, which means it's probably never going to be classified as a single peaked response. And then this one is a little bit more variable because we are getting to what looks like two distinct peaks here. However, because this trough does not drop below baseline, we're not calling it a single peaked response. So only in about 80% of recordings did that. In about 80% of recordings, that SOA gets called a single peaked response. So we can quantify the location of our peak, which is our peak SOA. So this is about 23 milliseconds. And then we can look at what we've defined as the neural window of integration. So this is the difference in our distribution at 70%. So what happens when we manipulate inhibition? So if we drop the level of inhibition, you can see we're drastically changing the profile that's occurring within the multisensory cortex. We're getting a single peak, but it's much later. We're only getting a single peak. And then we're getting what looks like one single peak. However, the second peak is occurring just much later. So if we plot this in our graphs, we can actually see that we're getting a shift in the relative timing of the audiovisual response profile, whereby it's becoming more visual, and we're actually getting a widening in our temporal window of neural integration, such that multisensory neurons are now losing their temporal sensitivity. So they tend to really prefer this timing, and now this is lost. So briefly, what happens at the level of perception? So again, 8 trained rats were used. And they were tested after an infusion of ACSF or gabazine. So we can see that following an infusion of gabazine, we're getting a shift in our PSS such that it's becoming more positive. So it's shifting in the visual first side. We actually do see an impairment in our temporal sensitivity. So this curve is primarily shifting on the visual first side. So an infusion of gabazine, or increasing the level of excitation within the brain, does shift the rat's perception of simultaneity, and it impairs their temporal sensitivity. And interestingly, this does primarily occur on the visual first side of perception, which is quite consistent with what we saw electrophysiologically in the brain on the previous slide. So to quickly summarize, do rats show perceptual malleability consistent with humans? Yes, we showed this on multiple facets, which means that future experiments can aim to uncover the neural basis of this perceptual malleability or these perceptual shifts. Using our translational models, we showed that the role of the multisensory cortex on audiovisual temporal perception appears to play a role in perceiving the relative timing of the audiovisual stimuli. And then when we manipulate the balance of EI within the V12 cortex, we see that, yes, it does greatly influence audiovisual perception and processing, which means that future experiments can investigate the cell types and neural circuits that are involved in the accurate perception of audiovisual stimuli. So with that, I'd quickly like to say thank you to my supervisor and some of the students that were involved in the lab, as well as our funding sources. And I'm happy to take any questions.
Amazing. That's great work you do there. Very interesting. Well, let's move to the questions part. So let me go through the chat first. Ashley, you have access to it, right? In case something comes up there. Okay. So I will give some time for questions to come up. I have a
No
question
problem.
myself.
Mhm.
You said maybe it's a naive one, but you said that you saw that the intensity of the stimulus affected the outcome of your results. So how do you interpret that finding?
How do I what, sorry?
How do you interpret that finding? How does the intensity affected the outcome?
Yeah, so we just played with the intensity of the acoustic stimulus. So basically what's happening is we believe that when we're decreasing the intensity of the stimulus, it's making it harder for the rats to hear the stimulus. So if they're not able to hear it, they're missing it. So then they're going to miscategorize it as a visual first stimulus. So that's why the curve is shifting in that direction, because they're missing the stimulus. They're like, oh, the auditory didn't happen. It was visual first. I'm going to categorize it as visual first. And then the opposite pattern is occurring when you're increasing the stimulus intensity. Because it's such a powerful acoustic stimulus, like 90 dB is quite loud. It's just so strong, it's overtaking, and you're going to categorize everything as auditory first. Because you're hearing it so loud and it's so dominant, you're then missing the visual and you're categorizing it more as an auditory stimulus.
Okay, so somehow it gathers the attention first. So I don't know, because my research is on humans. I don't know if the intensity of the sound is perceived as aversive from the rodents. So maybe it might not be only attentional, it might be arousal as well that plays a role there. I don't know.
It
I don't
might
have
be.
any clue about rodents.
Yeah, the rodents are they're very food motivated. So they're pretty they're very much paying attention to what's going on. And I would say their arousal level is pretty high. I would think the first few times they're presented with the louder stimulus intensity, it might be shocking the first few times, but I think they do get used to it pretty quickly.
Okay.
But they're very engaged in the task. So I wouldn't suspect arousal level would play too much of a role into it. I think it's more attentional and what they're picking up on.